Good morning, students. Today I'm going to take your class and I'm going to start with chapter 2. It's all about sexual reproduction in flowering plants, and under this chapter 2, sexual reproductions in flowering plants, you are going to study about pre-fertilization, the structures and events. Then under this chapter 2, you are going to study about fertilization, post-fertilization, the structures and events, pollinations. Then you are going to study about the special type of asexual reproductions. Okay, so here you are going to study all these things. And this morning I am going to start with the topic that is structure of anther. So here I have drawn a diagram, and this is the TS of anther. You all know what is TS. This is a transverse section of anther. Now, even in class 11, you have learned so much about the structure of flower. So, this morning we will start with the structure of anther. Now, what is anther? Anther is a part of stamens. As you all know, that stamen has got two parts, and that is anther and filament. Okay, and what is stamens? Stamens is a male reproductive organ of flower. So here, you have studied last year. This is the stamens, okay, and each stamen has got two parts, anther and filament. So this stamen is a male reproductive organ of a flower. Now, when you see anther, okay, each anther has got two lobes. Anther is bilobed, okay. Anther is bilobed, and each lobe, it has got two sacs. When you see under the microscope, the transverse section, so you will find that in each lobe, this is half lobe and this is another lobe, and in each lobe you will find two sacs, and that sac is called as pollen sac or microsporangia. Okay, and it is from this pollen sac or microsporangia pollen grains are produced. So what I said, each anther you will find that it is bilobed, two lobes are there. Okay, and in each lobe you will find two pollen sac or microsporangia, and in another lobe you will find another two sac or microsporangia. So altogether there are four pollen sacs or microsporangia. So this is 1, 2, 3, 4. Four pollen sac or microsporangia, and it is from this microsporangia where pollen grains are produced. So each lobe of anther, okay, the lobes of the anther, they are connected, okay, they are joined by a tissue called as connective tissue, and it has vascular tissues that help in carrying the nutrients. Okay, so as I said, that there are altogether four pollen sac or microsporangia. Now, when you see each microsporangium, it is surrounded by four walled layers. And what are those four walled layers? The first one is epidermis. Okay. The second one is the middle layer. I think I have not shown. Middle layer. Okay, it is surrounded by four walled layer. The first one is epidermis, second one is the middle layer, and next one is endothelium, and the fourth one is tapetum. Okay, a microsporangium is surrounded by four walled layer: epidermis, endothelium, middle layer, and tapetum. Out of this, epidermis endothecium and middle layer, they are the outermost wall layer. Okay, this tree layer is regarded as the outermost layer, and the main function is to protect. Okay, the main function is to provide protection. Okay, and it is from this outermost layer where pollen grains are released from the anther. Then we have another wall layer, that is tapetum, and this tapetum is the innermost wall layer. It is the innermost wall layer. And what is the function of this tapetum? It is to provide nourishment to the developing pollen grains. Okay, remember that it's very important to know what is the function of tapetum. So what is the function of tapetum? It is to provide nourishment to the developing pollen grain. And when you see the cell of tapetum, okay, which is the innermost wall layer, the cell of tapetum, it has got dense cytoplasm and it has more than one nucleus. Okay, so this is the different layers that is surrounding the microsporangium. Now, each microsporangium, as I said earlier, that there are 4 pollen sac or microsporangium. Each microsporangium, okay, it has numbers of sporogenous tissue or microsporocyte. Okay, each microsporangium, it has number of sporogenous tissue, or we even call as microsporocytes. Okay, each microsporangium, it has number of or various sporogenous cells or sporogenous tissues, or it is also called as microsporocytes, and it has dense or abundant cytoplasm with one prominent nucleus. Okay, so again I repeat, each microsporangium, it has compactly arranged microsporocytes or sporogenous tissue and has abundant cytoplasm with one prominent nucleus. Okay, and so that is the structure of the anther. Now, microsporogenesis. Okay, we'll start with microsporogenesis. Okay, here, before I come to that, I have not shown you this diagram. Here, this, as I've said earlier, that there are 4 pollen sac, 4 microsporangium, and when you view under the microscope, you will find this structure, microsporangium. Each microsporangium, the structures are in this manner. So this is the enlarged view of microsporangium. And this you can, here I have not completed the diagram. Okay, but when you see in your textbook, and when we try to view under the microscope, we will try to view in this manner. So you just look to your textbook and you try to complete the full diagram of microsporangium. So this is about the TS of anther. Now, microsporogenesis. Sporogenesis. What is this microsporogenesis? Now, microsporogenesis, it is the process. It is the process of formation of microspores, okay, from pollen mother cell. Formation of microspore from pollen mother cell, PMC, through meiosis. What type of cell division? It is meiosis. So the process of formation of microspores from pollen mother cell, it is called as microsporogenesis. So now, what happened? As I said earlier, These are the microsporangium. Now, what happened? Each microsporocyte, we'll try to study about the how formation is taking place, microspores or pollen grains. And for that, we have to know what happened here in the microsporocyte. So remember that each microsporocyte or sporogenous tissue, they undergo division, that is called as mitotic division, and they give rise to numbers of pollen mother cell or microspore mother cell. Okay? Microsporocyte or sporogenous tissue, each microsporocyte or sporogenous tissue, they will undergo mitotic division and it gives rise to pollen mother cells or microspore mother cells. Okay? And they undergo meiotic division. Now, this pollen mother cell or microspore mother cells, they are diploid in nature. They again undergo, another division to give rise to haploid microspores. Again I repeat, each microsporocyte, they undergo the mitotic divisions, and it gives rise to number of pollen mother cells or microspore mother cells, which are diploid in nature, and they again undergo another division and it gives rise to haploid microspores. And each microspores, it will give rise to pollen grains. So each microspores, again, what happened? They get separated out and they form thick outer wall. I'll just show here the microspores, what happened to the microspores. Each microspores will give rise to pollen grains. And this microspores, they get separated out as the anther get mature. As the anthers get mature, each microspores, they get separated and they form thick outer wall. They form thick outer wall, that is called as exine, and they form thin inner wall, it is called as intine. This is exine, the outer wall, it is exine, and the thin inner wall, it is called as intine. Again I repeat, as the anther get mature, as the anthers get mature, microspores, they get separated out, and they form thick outer wall called as exine and thin inner wall called as intine. And this is the structure of pollen grains. So always remember that in all the pollen grains, the structure is not same. The exine is not same. In some of the flower plants, the pollen grains may be star-like, spiky-like structure, the structure of exine. And intine is just a plain, but in some pollen grains of some flower, you will find without spiky-like structure. The exine is plain and this is intine. But one example, even in practical, if you have to conduct a practical, you are going to study pollen grain, the structure of pollen grains, and you are going to observe under the microscope each pollen grains. So you all have studied about one flower, that is Hibiscus rosa sinensis last year in class 11. And if you study the pollen grains of that flower, and observe under the microscope, you will find the structure of microspores or pollen grain are in this manner. The exine, the outer layer, the thick layer, you will find it is just a spiky-like structure, and the inner wall, the intine, it is in a plain form. So this is a structure of the microspore or pollen grain, which get separated out, okay, which are released from the anther as the anther get matured, and the nucleus. They undergo, okay, the meiotic divisions, and the nucleus, they give rise to two nuclei. The larger nuclei, it is called as vegetative nuclei or vegetative nucleus, and the smaller one, it is called as generative nucleus. Again I repeat, you need to know this. Single nucleus of this microspores, or each nucleus of microspores, previously it was only one nuclei, but they undergo certain division, and that single nuclei, it has divided into two nucleus. The larger one, it is called as vegetative nucleus, and the smaller one, it is called as generative nuclei. And what happened? Later on, this generative nuclei, okay, they will undergo division, mitotic division, and they will give rise to two male gametes. Okay, from this generative cells, they will give rise to two male gametes by the division of mitotic. Okay, by the mitotic divisions, the generative cells will undergo, they will divide, and they will give rise to two male gametes. And remember that this is also regarded as equivalent to male gametophyte. Okay, when you have to study how the development of male gametophyte is taking place. So we have to start from the structure of anther. Okay, and then we have to go with the different divisions, and finally you will find that the nucleus, which is undergoing division, giving rise to vegetative nucleus, generative nucleus, and it is from the generative nucleus, by certain division, they gave rise to male gamete, and this is the structure of complete pollen grains or microspores, and it is also regarded as equivalent to male gametophyte, the development of male gametophyte. So this is all about the structure of anther, as we are going to start with fertilizations, what happens to the this male gametes, what will happen to the vegetative nucleus, all this, okay, we will study step by step. But today I have only explained a little bit about the structure of anther, and now we will proceed in the next class what happened to this vegetative nuclei and where these two male gametes it goes. Okay, so for today I'll just stop here and we'll continue in the next class. Thank you.